Good evening, doctors. Biochemistry is a very scoring subject. It is very scoring. It may appear little boring, but it is scoring. You can make it more interesting. I will make it more interesting for you. So let us begin with the multiple choice questions. APO 48 is a remnant of A. RNA editing, B. RNA interference, C. RNA splicing, D. DNA translocation. A. RNA editing, B. RNA interference, C. RNA splicing, D. DNA translocation. ApoB48 is a remnant of A. RNA editing, B. RNA interference, C. RNA splicing, D. DNA translocation. DNA translocation. So, what do you think the correct answer here is? Let us go slowly. The correct answer here is RNA editing. Anybody? Why it is RNA editing? In liver cell, what happens? ApoB100 gene gives rise to ApoB100 protein. But ApoB48 protein is made in the intestinal cells from the gene encoding ApoB100 by the process of RNA editing, which is also known as differential RNA processing or chemical modification of RNA. This is a post transcriptional modification. RNA splicing is also post transcriptional modification, but this is removal of introns and not editing. RNA interference is silencing technique where the gene is sequenced by not letting the product of gene form. Translocation occurs during translation. This is a method of ribosome over mRNA. So the correct option here is RNA editing. Hepcidin decreases iron absorption by inhibiting. Hepcidin decreases iron absorption by inhibiting which enzyme? It is ferroportin. Hepcidin regulates iron metabolism. It mainly regulates iron entry into the circulation. By inhibiting ferroportin, hepcidin reduces the dietary iron absorption. The iron released from the macrophages. So increased hepcidin activity is responsible for reduced iron availability. Increased hepcidin will lead to anemia. Decreased hepcidin will lead to iron overload. Increased hepcidin will lead to anemia, and decreased hepcidin will lead to iron overload. Hepcidin synthesis and secretion by liver is controlled by iron stores within the macrophages, inflammation, hypoxia, and erythropoiesis. Correct option here is ferroportin. Which one of the following has highest glycemic index? It is glucose. Highest glycemic index. It is glucose. Glycemic index. This is the increase in blood glucose after the test dose of a carbohydrate compared with that of equivalent amount of glucose. Glycemic index is a value assigned to foods based on how slowly or how quickly the foods cause an increase in blood glucose levels. The highest glycemic index is for glucose and galactose. Also, lactose, maltose, trehalose, and isomaltose gets converted to glucose. So they also have high glycemic index. Glycemic index of fructose is less than that of glucose. Fructose and sugar alcohols have less glycemic index because they are not absorbed completely. Sucrose also has low glycemic index than glucose as it gets split into glucose and fructose. GLUT5 is a transporter for GLUT5 is a transporter for B fructose. GLUT5 is a transporter for fructose. Which one of the following is insulin dependent? Which one of the following is insulin dependent? GLUT4. GLUT4 is insulin dependent. Cause of HHH syndrome: hyperornithemia, hyperammonemia, homocystinuria. Hyperornithemia, hyperammonemia, homocystinuria is due to defect in ornithine transporter. Defect in ornithine transporter leads to hyperornithemia, hyperammonemia, homocystinuria. This prevents ornithine transport into mitochondria, and hence if the cytoplasm, that is hyperornithemia, and its reduced ability to clear carbamyl phosphate and ammonia, hyperammonemia. Mitochondrial carbamyl phosphate carbamylates lysine to homocysteine, leading to homocystinuria. So the correct answer here is defect in ornithine transporter. Which method is used to separate a mixture of lipids? It is chromatography. Wrinkled appearance of macrophage in bone marrow is Gaucher's disease. Correct answer is Gaucher's disease. Wrinkled paper or crumpled tissue paper appearance of macrophage in bone marrow aspirate is seen in Gaucher's disease. In CRISPR-Cas9 system, which repair mechanism is used for genome editing? CRISPR-Cas9 endonuclease system introduces double-strand breaks in the DNA at specific target site. Now, cell tries to repair this double-strand break by two mechanisms: non-homologous end repair or homologous repair. Non-homologous end repair mechanism is always a possibility as it does not require a homologous DNA. So it is a major repair mechanism during CRISPR-Cas9 system. The homologous repair will only work if the DNA homologous to cut DNA is present but is not always present. Chimeric DNA is used for organ transplantation. Rappaport-Leibig's cell shifts oxygen dissociation curve to right because of 2,3-DPG. Hot flashes are experienced as a result of increased noradrenaline and decreased estrogen. Restriction fragment length morphism is used for analysis of DNA fragment. Thermogenic food in which of the following does not depend on macronutrients? Ketone bodies are not utilized by brain. RBC. Enzyme deficient in hyperammonemia type 1 is carbamyl phosphate synthase 1. Type of mutation seen in sickle cell anemia is point mutation. Which replacement of amino acid does not alter its normal function? Glutamine replaced by asparagine. The clinical feature, it is pellagra, occurs due to deficiency of A. tryptophan. Which one of the following binds mRNA with riposome? It is 7-methylguanosine cap. Which of the following is anaplerotic reaction? Conversion of pyruvate to oxaloacetate. Which one of the following group of proteins assist in folding of other proteins? It is chaperones. Myoglobin has which structure? Myoglobin has primary, secondary, and tertiary structure. Best investigation for metabolic disorders is tandem mass spectroscopy. Separation of protein by their mass is done by gel filtration chromatography. So let us revise all the MCQs one by one. Apo48 is a remnant of RNA editing. Hepcidin decreases iron absorption by inhibiting ferroportin. Which one of the following has highest glycemic index? It is glucose. GLUT5 is a transporter for fructose. Which one of the following is insulin dependent? GLUT4. Cause of HHS syndrome is deficiency of ornithine transport. Which method is used to separate a mixture of liquids? Chromatography. Wrinkled vapor appearance, Gaucher's disease. Non-homologous hand repair. Chimeric organ transplantation. 2,3 DPG. Hot flushes are experienced as a result of increased noradrenaline and decreased estrogen. Ketone bodies are not utilized by RBCs. Enzyme deficient in hypertension CPS1. Point mutation. Tryptophan. 7-methicillin guanosine cap. Chaperones. Primary, secondary, tertiary. Tandem mass spectroscopy. Spectrometry. Gel filtration chromatography.